Hello everyone. Today I'll be explaining DNA fingerprinting, also called DNA profiling or your genetic fingerprint. Everyone except identical twins has a unique set of DNA. DNA is made up of four bases or letters, A, C, G, and T. These four letters form the written code that makes up the DNA sequence. Now, when someone says that everyone's DNA is unique, what they really mean is that when you pick two people at random and compare their DNA sequences, you will, on average, find a different base pair every 1,000 base pairs or so. This is where the statistic that says everyone's DNA is 99.9 the same comes from. Now, what DNA fingerprinting does is compare DNA samples from different people to analyze the differences in the remaining 0.1% and identify people at the molecular level. DNA fingerprinting can show relationships among family members, like in paternity testing, and is also used to solve crimes. DNA collected at crime scenes is used as evidence in many legal cases. DNA fingerprints can be made from a few cells because PCR or polymerase chain reaction can make a large sample of DNA even when there is a very small sample to start with. The greatest differences in DNA among people are found in regions of the genome that are not part of genes. As a result, DNA fingerprints focus on non-coding regions of DNA called short tandem repeats or STRs. STRs are stretches of 2 to 16 base pairs that are repeated many times. Each person's DNA differs in the numbers of copies of the repeats. For example, one person may have 8 repeats in one location and another person may have 10 in the same place. DNA fingerprinting uses special proteins called restriction enzymes to cut the DNA at certain locations where the STRs are found. Different restriction enzymes cut DNA at different places. Each has a unique sequence it recognizes. For example, the restriction enzyme EcoR1 cuts DNA at the sequence GAATTC and will cut only at that sequence. It will not, for example, cut at GACTTC. The differences in the number of repeats are found by separating the DNA fragments by gel electrophoresis. When there are more repeats, a DNA fragment is larger. In an agarose gel, smaller fragments run faster, so you get separation based on size. The bigger fragments are near the top, the smaller are near the bottom. Let's look at an example of how this might be done. Suppose Mary has a child, and she wants to determine which of two men, Bob or Larry, is the biological father of her child. She consults a paternity testing expert. The expert collects a certain stretch of DNA from Mary, Bob, Larry, and the child, and cuts the DNA with EcoR1. When the expert separates the cut DNA fragments on an agarose gel, the pattern looks like the one below. The child's DNA must be a combination of Mary's DNA plus one of the men's DNA. The agarose gel indicates that the child's DNA is a combination of Mary's DNA, top band, plus Larry's DNA, bottom 3 bands. Thus, Larry is the biological father of the child. What happens if Larry and Bob have the identical sequence in this stretch of DNA? The answer is that you wouldn't be able to distinguish based on looking at differences in this stretch of DNA between the two men. So what do? You simply search for other stretches of DNA in which there is a difference between these two men. This is why in real life, multiple stretches of DNA must be examined to ensure